The title of this presentation is Gap Junction Remodeling in the Atrial Fibrillation Arrhythmogenic Substrate. First, let me define what arrhythmogenic substrate means. It's the electrophysiological environment that promotes or enables arrhythmogenesis. And this environment is composed of a number of different components. Sarcolemmal ion channels of myocytes, gap junctions which connect myocytes to each other, and myocardial structure which can be altered in the arrhythmogenic substrate. And all of these affect impulse initiation, conduction, and refractoriness. Before speaking about gap junction remodeling, it's beneficial to just quickly review the normal location and function of gap junctions. Gap junctions are channels that connect myocytes, adjacent myocytes. In each of the adjacent myocytes, there's a partial junction called a connexon, and two opposing connexons form the gap junction channel. The protein that makes up the channel is called connexin, and there are a number of different connexins in the heart, named according to their molecular weight in Daltons. And the function of these channels is to allow the passage of small molecules and ions from one cell to the other, and we're particularly concerned with the passage of ions because that's what controls electrical activity. In the atria, there are several different connexins, connexin 43 and connexin 40, unlike the ventricles where there is only connexin 45. And these connexins are located at the ends of the cells, as shown here by this line. This is immunofluorescent staining for connexin, and they're mostly located at the ends of the myocytes. One of the reasons that we're concerned with the fact that there are several different connexins in the atria rather than only one is that the different connexins can connect with each other, as shown here, connexin 43 to connexin 43, which is homotypic, or connexin 40 to connexin 40, or connexin 43 to connexin 40. And all these combinations have different ion channel conductances, which makes it very complicated to figure out how remodeling of connexin affects propagation of the impulse. So what is gap junction remodeling? We can define gap junction remodeling as a change, usually a decrease in coupling between cells, and it is often caused by a change in conductance of the gap junction ion channel. This can happen without any changes in amount or location of the channel. It can also occur as a change in the quantity of the connexin, and this also changes coupling, and a change in the location of the connexin, and usually it shifts to lateral membranes, whereas in the normal heart it's located only at the ends of the cells in the intercalated disc. So let's look at a change in coupling and remodeling without a change in the amount or location of connexin. This is best demonstrated by the effects of low pH on the connexin channel conductance. And this example is shown here in oocytes with expressed connexin 43, not one of the primary connexins of the atria, but still important for the atria. And these oocytes were exposed to a low pH, as shown here, and as a result, the conductance, or the ability of the gap junction channel to pass current, decreases quite quickly. The model is shown over here. The gap junction channel is pictured as a channel with a ball and chain mechanism for closure, which is the carboxy terminal of the connexin. And pH causes this to swing over across the channel to decrease the ability of ions to pass through it. More commonly, the change in the amount of connexin and lateralization occurs in the atrial fibrillation arrhythmogenic substrate. This shows data from human tissue from Costin et al., who measured connexin 43 in post-op specimens from the atria, and you can see that the amount of connexin 43 is markedly reduced in the right atrium in patients with atrial fibrillation. Also, this is the appendage, markedly reduced in the free wall in patients with atrial fibrillation. And likewise, the other primary connexin 40, is reduced in these patients with atrial fibrillation, as shown here. If we look at staining, immunostaining for connexin 43 in atrial tissue from patients with atrial fibrillation, you can see in the right atrium from normal patients that the immunostaining, shown as yellow, is at the ends of the cells, primarily at the ends of the cells, and you can get a feeling for the amount of connexin 43. And here, in patients with atrial fibrillation, you can see there's a reduction in the amount of staining, which indicates a reduction in connexin protein or gap junctions, and also location of connexin along the lateral membranes. This also happens with connexin 40. So both connexin 43 and connexin 40 can decrease in amount, as shown here. It's heterogeneous in that there are regions with a lot of connexin and regions without connexin, and there's also lateralization. And the other thing that affects gap junction connections in patients with atrial fibrillation, particularly long-standing atrial fibrillation and structural heart disease, is the large amount of fibrosis that occurs in the atria, as indicated here by the red staining, which separates myocyte bundles and pulls them apart physically so that there are fewer gap junction connections between these myocytes. So how might gap junction remodeling contribute to atrial fibrillation? Well, first let's look at what do we think are the mechanisms for atrial fibrillation, and there are a number of different possible mechanisms. The primary mechanism for atrial fibrillation involves reentrant excitation, where a wavefront moves around and around in circles, and the ability of the wavefront to move around in circles is dependent on the wavelength. And the wavelength is a product of the refractory period and the conduction velocity. So anything that decreases either refractory period or conduction velocity facilitates reentrant excitation. In a normal-sized atria, as shown here, there may be only room for one or two reentrant circuits. But when you have a large atria or a short wavelength, you can get multiple reentrant circuits. And this is facilitated, as I said before, by a decrease in conduction velocity and a decrease in the refractory period. So a decrease in conduction velocity is very important to get reentrant excitation to cause atrial fibrillation. The other current theory about causes of atrial fibrillation is the location of what is called a driver in the vicinity of the pulmonary veins, which then emits impulses with a very rapid rate into the rest of the atria. So